DIA is a multidisciplinary professional association that brings together regulatory agencies, the healthcare industry, academia, and patient support groups from around the world to advance the development and life cycle management of safe and effective medicinal products and to improve global healthcare by increasing access to these products. DIA has 5 decades of experience providing a neutral forum for discussing critical issues, challenges, and opportunities related to healthcare innovation. DIA is advancing conversations surrounding comparative effectiveness research and health technology assessment, global regulatory capacity, orphan drugs, biotechnology, medical devices, supply chain management, safety, and the development and delivery of vaccinations and other regulated products to populations around the world. We are a community advancing healthcare innovation to expand access to the most effective and affordable patient care. As a member of this community, you are an integral part of our future in improving health and well-being worldwide.
女士们、先生们，掌声欢迎 DIA 中国区总监蔡一智博士向各位介绍本届大会嘉宾。
尊敬的领导、嘉宾、女士们、先生们、朋友们，大家好！欢迎大家参加第4届 DIA 中国年会，很高兴能在上海和大家相见，会见很多老朋友，也结识很多新朋友。DIA 年会是 DIA 中国与中国医药国际交流中心共同举办的会议，已经成功的举办了第3届。本次第4届年会近1000人参加。我借此机会代表大会主委会对各国政府部门、科研机构、制药企业、临床医院和基地、大学院校及制药相关服务机构的积极参与，表示衷心的感谢。向 DIA 中国顾问委员会全体成员、所有支持本届大会的演讲者、专家及所有工作人员及志愿者表示感谢。向本届年会的所有参会者表示衷心的感谢。感谢大家为实现今年年会的目标、合作与创新而共同努力。现在请允许我介绍出席本次年会的主要嘉宾。国家食品药品监督管理局局长尹力博士，此次国家食品药品监督管理局参加这个年会的还有国际合作师许佑军师长、药品注册师师长张伟、信息办主任孙贤哲、国际合作师副巡视员秦小颖、中国药学会秘书长丁立霞、国家食品药品监督管理局药品审评中心副主任张培培。今天出席开幕式的 DIA 领导有 DIA 全球主席 Eve Julian 博士、DIA 全球执行总监 Paul Pomerantz 先生、DIA 后任主席舒岭博士。本届年会的大会主席 和副主席胡江斌博士，赵雅君主任，韩颖史博士。我也很荣幸向大家介绍在前边就座的大会演讲的一些嘉宾以及其他的嘉宾，他们是国际著名学者、艾滋病鸡尾酒疗法发明者何大一教授，Plenary 公司副总裁 Alberto Gignolo 博士，欧洲理事会药品质量局办公室局长 Susan Patel 博士，美国商务部消费保健品办公室主任 Jeff Green 先生，英国药品医疗产品监管局 GCP 部门负责人 Vincent Young 先生，新加坡卫生科学局应用科学部化学与物理实验室主任君令。除此，我们还有很多嘉宾，来自加拿大卫生部的、日本 PMDA 及美国 FDA 的代表、WHO 的代表，还有呢，就是我们许许多多的这些专家和学者们，从全世界各地来参加 DI 的年会，让我们用热烈的掌声欢迎我们所有嘉宾。下面有请 DIA 全球执行总监 Paul Plenary 先生致开幕词，谢谢大家。
Ni hao, and welcome to our distinguished guests, speakers, and attendees. This fourth annual China meeting is DIA's largest in China to date. DIA has been active in China since 2008 with a high-level advisory council as well as a professionally staffed office in Beijing. China represents DIA's most rapidly growing region. Over 30,000 people annually participate in DIA programs. We have a membership of approximately 18,000. We are active in 6 regions of the world: China, India, Japan, Europe, North America, and DIA's newest, Latin America. Each region is guided by a volunteer advisory council and supported by a dedicated professional staff. DIA is known for its conferences, training programs, our publications, and our 30 professional communities. It is fitting that this energetic meeting be held in Shanghai, one of the world's most vibrant cities. It is a city that is modern, innovative, and rapidly growing. So is the case with DIA. Under the leadership of our president, Dr. Yves Gillet, and the board of directors, DIA is evolving to serve the changing healthcare needs of our regions and the world. DIA continues to develop globally in content, programs, in regions, and in addressing global health concerns. In 1 month, Dr. Ling Su will take office as DIA's first president from Asia. Dr. Su's election is a testament to his leadership in China, his many accomplishments, and the importance of China globally to medicines. We encourage you to become engaged in DIA, to take advantage of what we have to offer, and to contribute your own suggestions, your talents, and expertise. Please visit, during this conference, our DIA booth to learn more. Our booth is on the exhibition floor on the first floor. As with the meeting, the exposition over the years has continued to grow, and we appreciate the contribution it makes to this overall experience. It is now over 50 booths, and we encourage you to spend time with our exhibitors. It is an excellent opportunity for networking in between educational sessions. The extent of this program is a testament not only to the hard work of our volunteers and our speakers, but also to DIA's dedicated staff and service providers. I'd like to take a moment to thank Dr. Jane Kai and her outstanding team for their hard work in producing this excellent event. I will be here through the full conference and look forward to meeting with as many of you as possible. My best wishes to all of you for an excellent and successful conference. Thank you.
女士们、先生们，掌声欢迎中国国家食品药品监督管理局尹力局长为本届大会致辞。
尊敬的各位同事、女士们、先生们，首先，我代表中国国家食品药品监督管理局，对第4届药物信息协会中国年会的召开表示热烈的祝贺，向出席会议的各国药品监管机构、研发机构、生产企业和学术界嘉宾表示诚挚的欢迎。近年来，全球化、信息化快速发展，药品不仅与公众健康和生命安全密切相关，而且与国民经济和社会发展密切相关。药品领域的合作与创新越来越重要。本次年会围绕着合作与创新，共同探讨中国医药以及世界医药的未来发展，很有意义。中国药品监管部门十分重视，致力于推动医药领域的合作与创新。多年来，在坚持加强药品安全监管、完善药品生产供应体系的同时，积极开展与世界各国和地区的战略合作，密切保持与国际组织或地区组织的联系，在促进药品经济健康发展、提高社会公众健康水平方面做了大量的工作，取得了一定的成绩。一是药品安全监管能力不断提高，中国药品法律法规体系和标准管理体系日趋完善，质量管理进一步规范，国家、省、市、县4级药品监管工作不断加强，基础设施明显改善，技术队伍素质明显提高，药品监管措施不断的丰富，全品种药品电子监管顺利推行，国家药品储备制度建立，应对重大疫情灾害的药品保障能力进一步的提高。二是公众用药的需求得到了较好的满足，药品安全状况逐步改善，药品质量总体上保持较好的水平，公众用药安全权益更加有保障，国家基本药物制度进一步的实施，公众基本用药权益也得到了保障。通过政策引导、标准引领，新药创制能力不断提高，药品现代物流体系建设稳步的推进，覆盖城乡的药品供应网络 基本建成，公众日常用药的需求基本得到了满足。三是医药经济实现了持续快速发展，中国医药工业体系更加完备，医药产品更加丰富。目前，中国已成为世界原料药生产大国，生产的化学原料药以及中间体达到1500多个品种，产量约占世界原料药市场份额的22%。青霉素工业盐、维生素 C、红霉素、四环素等发酵抗生素初级产品的产量占世界第1位。2011年，中国医药经济持续保持平稳较快的发展势头，医药外贸进出口总额实现了733亿美元，同比增长39%。医药工业总产值达到1.5亿元，同比增长23%。在看到成绩的同时，我们也清醒地看到，在发展中也存在着一些问题，监管工作面临着巨大的挑战。从国内看，医药企业发展不平衡，差异较大，多、小、散、乱、差的问题仍待解决。监管体系也有进一步需要完善的地方。最近，葛超标药用胶囊事件也给我们敲响了警钟。从国际看，世界各国健康需求持续增长，全球化医药市场融合加快，药品研发、临床试验、生产销售和消费使用，在网络信息技术的推动下，已经打破了国界。这些变化要求我们在研发创新上要做更多的工作，要在监管上整体的提高我们的能力。面对新的形势、新的任务和新的问题，中国政府在国家药品“十二五”发展规划中明确提出，要经过5年的努力，实现药品监管体系进一步完善，药品研制、生产、流通秩序和使用行为进一步规范，药品标准和药品质量大幅度提高，药品安全保障能力整体上要接近国际水平，药品安全水平和人民群众用药的安全满意度要显著提升。我们有信心、有能力解决好发展过程中出现的各种问题。女士们、先生们、朋友们，广泛加强合作，致力推动创新，是抓住机遇、应对风险、解决问题、实现发展的必然选择。中国药品监管部门希望同世界各国同仁共同努力，实现4个美好的愿景：一、提高能力，应对药品安全风险。中国药品监管部门将提高监管能力作为不懈的追求。未来几年将努力做到3个方面的工作：一是加快实施电子监管，为保证药品生产流通全程质量可控、可追溯，我们启动了药品电子监管制度的建设，对药品分步实施电子监管码管理，目前已完成了 麻醉药品、精神药品、血液制品、疫苗、中药注射剂等高风险品种的电子监管，正在推进实施国家基本药物全品种的电子监管。我们的目标是，到2015年底，对所有上市药品实施电子监管。这将对追溯产品的流向、保障产品的质量安全、提升监管水平发挥重要的作用。二是全面推行新版药品 GMP。中国药监部门吸收国际先进的理念和技术，对药品 GMP 进行了修订。未来几年，我们将分步实施新版药品 GMP。2013年年底前，使血液制品、疫苗、注射剂等无菌药品生产企业达到新版药品 GMP 的要求。到2015年底前，其他类别的药品生产全部达到新版药品 GMP 的要求。中国药品监管部门将坚定不移地推进实施这项制度，在规定的限期内，没有达到要求的企业还有时间，将坚决地停产。三是努力提高技术能力，重点是加快推进药品快速检验技术在市场监督中，在市场的监管一线和基层地区的应用，提升药品的检验检测的能力，加强药品不良反应的监测，完善上市后再评价，加强安全风险预警体系建设，提高突发事件的应急处置能力。二，推动创新，提升药品安全保障水平。中国药品监管部门充分发挥政策、技术和标准的作用，加快药品研发创新，促进药品质量提高。一是加强政策引导，中国政府已经将生物产业作为加快转变经济发展方式、引导经济科学发展的战略性新兴产业，同时将继续实施国家重大新药创制战略，探索完善以药物自主创新能力提高为目标，导向明确的审评机制与策略，围绕危害国民健康的重大疾病，顺应对重大公共卫生事件的需要，努力培育一批具有自主产权和国内外重大市场前景的创新药。二是加强药品研制的管理，建立研究机构与注册品种核查相结合的工作创新制度和临床实验机构分级分类管理的工作机制，提升临床试验实施水平和管理水平，加强审评与认证检查、监测评价、检验检测、标准管理等环节的沟通和配合，提高创新药和临床申请的审评效率。三是强化标准引导，完善标准管理制度，加快建立标准评估和淘汰机制，努力提高标准工作的国际化水平。中国药品安全“十二五”规划对药品的标准提高提出来了要求，这就是 实现化学药品、生物制品标准与国际标准的接轨。中药标准能够引领国际标准的制定，医疗器械的国际标准采用率和转化率有明显的提高。三、加强合作，共同维护公众用药权益。伴随全球化进程，药品安全不是一时一地的问题，各国政府、监管部门、研发机构和医药企业都应具有时代的责任感和使命感，主动承担起维护公众用药权益的职责。中国药品监管部门的态度是，愿意同各国、国际组织以及民间机构一起，为共同保障药品的安全、维护公众用药权益而努力。我们将进一步完善监管制度和规则，提高监管标准和技术水平，深入继续的开展药物研发技术和药品监管经验、人才培养等方面的交流与合作，引进国外高端的检验检测、监测评价技术和方法，坚定不移地推动医药市场的对外开放，大力实施引进来和走出去的战略，支持引导国内医药企业参与世界市场的竞争，接受国际认证机构的检查，提高药品安全的保障水平。四、实现共赢，推动医药产业可持续发展。中国医药卫生体制改革正在全面的推进，基本药物制度的实施将进一步提高医药产业的集中度。中国药品监管部门将配合产业发展政策，加快制定和实施扶优劣汰、促进集中的监管政策，健全市场退出机制，引导产业增强核心竞争力。据预测，未来的10年，世界的药品市场年增长率将达6%到7%，中国药品市场的规模增长率将远高于世界的平均增长率。中国医药市场巨大，医药产业正处上升期，世界各国企业、研发机构都可以从中得到良好的发展机遇。中国药品监管部门愿意同世界各国同仁一起，在实现自身发展的同时，推动中国乃至世界医药产业的可持续发展。女士们、先生们、朋友们，世界各国的联系越来越密切，让我们进一步的加强合作，推动创新，共同为实现促进世界人民的健康和医药产业的可持续的健康发展贡献我们的力量。谢谢大家。
女士们、先生们，掌声欢迎 DIA 主席 Eve Juliet 博士为本届大会致辞。
下午好。Dear Mr. Commissioner, ladies and gentlemen, good afternoon. On behalf of the global DIA member and volunteer community, who I serve as president, please allow me to welcome you Shanghai to this DIA's 4th China annual meeting. The practice of healthcare has for centuries been one of the Chinese most recognized cultural traditions. DIA is honored and pleased to participate in the continued advancement of this historical tradition. Since our inceptions, nearly 50 years ago, DIA has created educational and networking forums that bring together the best and brightest thought leaders from government, industry, academia, and scientific and professional associations to collaboratively discuss new ideas and best practices that will advance the health and well-being of patients around the world. I would like to take this opportunity to thank all distinguished guests, well-known experts and participants. I would like to thank most especially Dr. Yin Li, the Vice Minister of Public Health of China, the Commissioner of State and Food Administration, for taking time from his very busy schedule to attend this event. This clearly demonstrates the Chinese government's commitment to foster innovation. I also thank our keynote speakers, Dr. David Ho and Dr. Alberto Grignolo. I'm very pleased to see this meeting is a reflection of the cooperating spirit for which DIA continues pursuing for the global health benefit. I extend my sincere congratulations to the program chairpersons, Mr. Zhao Yao and Dr. John Yu, the program committee, the DIA China Advisory Committee, ACC, and its chair, Lin Chu, an open and direct dialogue with CDE. I will personally explore all these educational and networking opportunities while participating in this meeting, including the products, services, and technologies displayed by the wide variety of domestic and international exhibitors. And I invite you to join me and to do the same. We look forward to sharing our complementary perspectives and experiences with each other as we come together to collectively pursue this meeting theme, collaboration and innovation in China. My best wishes for a wonderful educational and networking experience here in Shanghai. And thank you all for participating in this 4th China annual meeting. 祝贺大会成功，谢谢。
女士们、先生们，掌声欢迎本届大会联席主席胡江斌博士为本届大会致辞。
下午好，尊敬的国家食品药品监督管理局尹力局长，尊敬的 DIA 全球协会主席易Julian博士。首先，我谨代表年会主委会热烈欢迎各位参加第4届 DIA 中国年会。今年 DIA 是首次在中国的金融和经济中心上海召开这么一个年会。上海也是中国乃至世界上新药创新开发的重镇之一。大会的主题“合作与创新”点明了当今生命科学和健康产业发展的主旋律。只有不断的创新，才能推动产业的发展。只有携手全球的科学家、企业家和政府监管部门共同合作，我们才能开创健康产业的新的未来。在今年的大会上，我们非常荣幸地邀请到何大一医学博士和 Alberto Grignolo 博士做主题发言。何大一是当今世界上对艾滋病毒疫苗研究最顶尖的科学家。他于15年前对 HIV 病毒的研究开创了著名的鸡尾酒疗法。 使得艾滋病人的死亡率在发达国家自1996年以来下降了600%。他的工作拯救了成千上万人的生命。第二位演讲嘉宾 Greg Nono 曾任美国药物法律事务协会董事会主席。今天他将与我们分享他从事新药开发30多年的经验和对新药开发的战略思考。除此之外，我们这次大会也增加了不少新的亮点，比如说，国家新药审评中心 CDE 将举办一个经验分享咨询会，这是 CDE 第一次以互动的形式在 DIA 大会上与参会者做近距离的交流。此外，美国商务部和药检局的国科师和注册师也利用 DIA 这个年会的平台，成功举办了一个关于原料药质量方面的研讨会。我们还非常荣幸地请到了我们的近邻以及世界上第2大医药市场的日本的 PMDA， 这次专门举办一个分会，介绍日本最新的药品注册的法规。此外，我们这次年会还第一次增加了有关疫苗、生物制药、合同外包 CRO 以及高新药股产业园区的专场报告会。最后，我想代表大会组委会借此机会，感谢我们的专家志愿团队、DIA 办公室的全体员工以及参展商，并预祝大会圆满成功。谢谢。
女士们、先生们，掌声欢迎本届大会联席主席赵亚军主任为本届大会致辞。
尊敬的尹局长，尊敬的一副主席，各位嘉宾，女士们、先生们，下午好。由中国医药国际交流中心和美国 DIA 共同举办的第4届 DIA 中国年会，今年移师上海，在中国这个最现代化也最繁荣的都市召开，希望能给各位嘉宾带来不同于往届的感受，能从全新的角度感受到今年的年会的主题：中国与世界，合作与创新。当今的世界，随着科学技术的不断发展，创新药物研发全球化的发展大趋势，对医药产业提出了更高的要求。面对这样的大趋势，机遇与挑战并存，我们必须加倍努力，提升新药研发能力，积极参与国际竞争，带动国内新药创新能力水平的提高，努力使我们的监管政策、技术要求逐步与国际接轨，把创新药物推向一个更新、更高的层次，从而使新药研发产生根本的变革。在本次为期3天的会议中，来自国内外政府部门、学术机构和制药界的专家、学者共聚一堂，各抒己见，畅所欲言，针对新药研发、临床研究、GMP、药物安全、监管政策和数据管理等话题进行深入的经验交流，分享研究成果。同时，会议还特别增设了药品技术审评专题，届时大家有机会与药品审评中心的领导和专家们面对面的交流与沟通大家感兴趣的话题。在此，我仅代表会议主办方向出席会议的各位领导、嘉宾、国内外同仁、各位专家和企业界的朋友们表示热烈的欢迎和诚挚的感谢。中国医药国际交流中心一直致力于中国药品监管事业的发展，不断拓展合作的领域，深化合作的内容，加强国际合作与交流。从2009年开始，我们与美国药物信息协会合作举办 DIA 中国年会以来，搭建起了药品监管机构、科研机构、企业界和学术界的国际交流平台，希望通过技术创新、药物研发的经验交流，提升新药研发的能力，努力使我们的法规、技术要求 逐步与国际接轨，为促进药物的研发全球化，推进中国医药产业更加深入地参与国际竞争与合作，起到了积极的作用。我们应紧紧抓住培育和发展战略期新兴产业的机遇，借助深化医药卫生体制改革，推进实施基本药物制度的东风，发挥好大会的平台和桥梁作用，在加强国际合作，及时跟踪和掌握世界医药研发技术及相关领域的发展前沿，促进医药经济健康发展，鼓励支持创新药物的研发，为保障人民的用药安全做出我们的贡献。让我们通力合作，共同努力，肩挑民众健康，心系社会责任，不断创新，为医药研发的快速发展，为加快推进药品监管目标国际化的战略实施而努力奋斗。谢谢大家。
感谢赵亚军主任为本届大会致辞。EIA 中国区顾问委员会主席徐宁博士为本届大会颁奖。
So then, again, for tradition with the EIA is to pay tribute to the key volunteers, and we have this kind of ceremony in the different annual meetings, and it is the second time that here in China we have this award ceremony. So I'm here with Ning Hu, who is the chair of the advisory committee EIA China, and Jen Kei, who is in charge of the EIA here in China. So we'll have the pleasure to have two awardees today for outstanding service with the EIA. So the first, I'm not to destroy the mic. So the first awardee is William Wang. So Dr. William Wang, I will shortly summarize his bio. He's the head for the Department of Biostatistics and Research Decision Science, Asia Pacific Regions Node Research Laboratories. Dr. Wu has over 17 years of experience in the pharmaceutical industry with expertise in statistical design and analysis, scientific programming, and clinical data management. In 2007, he moved to China to establish a Merck's biometrics operations in support of MRL, local, regional, and global clinical development. So since 2007, he has initiated and co-organized multiple international biopharmaceutical statistics conferences in China. And in the last 2 years, he helped organize the DIA China's popular biostatistics and data management workshops, and served on the DIA China Regional Advisory Committee. And most recently, he was nominated to join the DIA SAIC Leadership Council, SLC, which is a very important committee. You know, the SAIC is a group of community of people working in the same area and working together. This is a way of being member of the DIA, is to be able to participate in the SAIC. And in that, he will promote the DIA global SAIC initiatives in SAIC. So I'm really pleased to welcome you and to give you this award, which was given by Nico and James. So the second awardee is Dr. Daniel Liu. Dr. Daniel Liu, if you could come. So Dr. Daniel Liu is currently he works as Director of China Development at Medidata Solutions Worldwide. He is receiving PC in Pharmacy and Master of Science in Pharmaceutical Chemistry from China Pharmaceutical University in Nanjing. And he got his PhD degree in pharmacology from University of Illinois, USA. And he had more than 10 years of researching experience in academic environment before joining different companies. So he has been involved in DIA in many ways, and particularly in working in the pre-conference workshops at DIA China annual conferences in 2009, 10, 11, and 12, which is quite a lot. He is also a member of the advisory committee, and in 2011 he successfully initiated and chaired the first DIA China data management workshop in China. So congratulations and thank you for DIA on behalf of DIA.
请我们再次用热烈的掌声祝贺两位获奖者。女士们、先生们，掌声欢迎胡江斌博士为现场来宾介绍本次大会演讲者。
下面我很荣幸地介绍我们今天大会主题发言 Keynote Speaking 的第一位演讲者何大一。何大一，如果说起何大一，可能我们在座的所有的这个中国人或者华人对这个名字都应该是不陌生的。15年前他所发明的这个鸡尾酒疗法，对这个艾滋病的这个治疗是一场革命性的贡献。何医生他现在的正式职位是美国纽约洛克菲勒大学艾滋病研究中心主任。他是在1974年在加州理工学院获得学士学位，在1978年哈佛医学院取得了医学博士学位，然后在取得博士学位以后呢，先后在美国加州大学洛杉矶分校UCLA 和美国麻省总医院以及哈佛医学院进行了博士后的研究。那么近年来呢，可以说由于以何医生为代表的这一些这个艾滋病的研究者的卓出贡献，目前这个艾滋病的成活率在西方发达国家已经是大大的延长了。所以说谈奈变色这个说法可以说是一去不复返了。如果我们以5年或者10年的成活率来算，现在这个艾滋病的成活率可以说基本上已经是远远超过了任何癌症的这个成活率。那么这些的工作都以何医生以及他的团队以及所有从事这个行业的贡献所分不开的。近年来呢，何医生的这个工作呢也逐渐从治疗艾滋病的治疗转向了艾滋病的预防。那么他现在呢领导的团队呢正是在主要从事疫苗艾滋病疫苗方面的研究。那么呢同时呢他还领导了一个由中美2国科学家参加的关于艾滋病疫苗的这么一个联合工作组。由于何医生杰出的贡献，他先后在全球12所大学取得了荣誉博士学位，并且呢在国内他是北京协和医学院、中国医学科学院、中国科学院、香港大学、武汉大学、复旦大学等等的客座教授。然后他也是麻省理工学院的董事会成员。在1996年，何医生被著名的《时代》杂志 Times Magazine 评为年度风云人物，并于2001年获得美国总统奖。所以说，我们可以说，何大一，你的工作和贡献再次提醒我们今天在座所有的医药人，我们所面临的肩上的重任和我们每天辛苦工作的意义。So without further ado.
Dr. David Ho.
Thank you very much, John, for that nice introduction. Good afternoon, ladies and gentlemen, colleagues and friends. I had the pleasure of participating in the DIA conference in Chicago last year, and it is likewise a pleasure and honor to be able to participate in the DIA China 4th conference. I'm going to speak to you about a project that is ongoing in the field of HIV prevention. Well, the slides are there. It is a project that's focused on the use of a particular antibody as a passive immunization approach to HIV prevention. And as it turns out, that this is a project that has ongoing collaboration between organizations in the US where I work and organizations in China where many of my collaborators work. Needless to say, HIV/AIDS epidemic is one of the worst in human history. The causative virus, HIV, has already killed about 25 million people worldwide, and about 33 million are living with this infection, which would be lethal in 99% unless treated. And we are all well aware of the devastation in places particularly like sub-Saharan Africa. Even though, as Dr. Hu has mentioned, even though HIV is now considered a treatable disease in developed countries, we must bear in mind that this epidemic continues to rage with about 2.5 million new infections each year. So we desperately need a vaccine that would slow or halt the spread of this virus. And yet, no such protective vaccine is available today, and in my opinion and in the opinion of others in the field, we are unlikely to have a vaccine in the near future. As all of you are aware of, there are a number of clinical trials that are ongoing, but they are mostly early-stage trials. A number of late-stage trials have been conducted, but all but one have failed. We are well aware of the failure of protein vaccines based on the envelope protein of HIV. We're well aware of the failure of vaccines such as one that was made by Merck using a viral vector. The only success is rather modest. It utilizes 2 different vaccines in combination, and yet each one of those had failed in separate trials. But the combination of the 2 in a trial conducted in Thailand called RV144 yielded modest protection of approximately 30%. Exactly how that protection is conferred is not known, and obviously it is worthy of additional studies. But practically speaking, those 2 vaccines together will not constitute a vaccine that would be amenable to widespread use. So it is safe to say that we are struggling to come up with a useful vaccination strategy. And the reason is HIV. HIV is very refractory to antibody neutralization. And if you look at this slide, it is mostly depicting the top view of the HIV external glycoprotein. As you could see, it is a symmetrical trimer with 3 units of protein coming to form a single spike on the virus surface. It has many variable sequences, and hence this virus could obviously vary from strain to strain, and therefore it is very difficult for antibodies to recognize a common determinant shared by all strains of HIV. In addition, as you'll see from the little sticks coating the outside of the spike, these consist of sugar or glycosylation. And there is extensive glycosylation so that half of the molecular weight of this protein consists of sugar. And the sugar, in fact, serves as a shield to protect the critical determinants on the envelope protein. Better depicted on this additional slide. So if you look at from the left to right, there are the core elements consist of the protein, but you started to add the known sugar residues on HIV, you see the addition of the blue, so that many of the critical components, such as one shown in yellow, required for receptor binding, are well shielded by the sugar moieties, and hence the antibodies find it much more difficult to penetrate this particular shield to bind to the functional elements. So a major reason we do not have a vaccine for HIV is because of the structural obstacles posed by variable sequences as well as the glycosylation. However, despite the lack of vaccine success, there are significant advances in the field of HIV prevention, and some of which are listed on this particular slide. The vaccine trial RV144 that I mentioned earlier is shown in green, and you could see the efficacy is rather modest, around 30%. A number of trials, particularly done in sub-Saharan Africa, using male circumcision has shown protection to about 50–60%. And obviously that is being implemented particularly in Africa. Quite recently, several studies pointed out that if you treat the infected person very, very well with combination antiretroviral therapy, you will help protect his or her sexual partner to an efficacy greater than 90%. And this, in our field, is known as treatment as prevention. And those results are reflected at the top of this graph in red color. But an area that has gained a great deal of traction is shown in purple as PrEP, which stands for pre-exposure prophylaxis using antiretroviral drugs. And you could see 4 separate trials done in America and in Africa have shown that the efficacy could range from about 40 to 70%, with the person who is at risk for HIV infection taking generally daily pills that target HIV reverse transcriptase. And just a week ago, an FDA panel approved a combination drug called Truvada for use, or recommended this combination pill for use as a preventive among American homosexual men who are at particular high risk for HIV infection. But you will also note that there are additional studies that fail to demonstrate protective efficacy. And to make a very long story short, the failure or the lack of complete success is largely due to patient adherence or subject adherence, in that it is very difficult to take medications on a daily basis for indefinite period of time when, in particular, one is still feeling well. So as a strategy, we could think about the next generation of PrEP, which could be infrequently administered. And this is a concept that my group has been pursuing, particularly thinking about monoclonal antibodies that are indeed capable of blocking HIV infection. And as you know, antibodies are administered less frequently and in general have a pretty safe profile. This is a slide that shows the breadth and potency of some of the existing HIV neutralizing monoclonal antibodies. Until about 3 years ago, all we're looking at are the 4 antibodies listed on the left side of this graph. And as you could see, under breadth, they have limited breadth, and potency as reflected by the inhibitory 50% inhibitory concentration, or IC50. These initial, what we call first-generation antibodies, lack both potency and breadth. But in the last 3 years, this landscape has improved a great deal. And a whole array of antibodies have emerged. And now you could see they have breadth approaching 90% and potency approaching concentrations that are actually achievable on a clinical basis. So you will see in the coming years a great deal of movement in the area of using such antibodies for HIV prevention. And these antibodies target certain critical domains on the HIV envelope spike. For example, in green is the region that is responsible for binding to the receptor on the cell that is susceptible to HIV infection, and that those would be CD4 positive cells. And in red, another epitope, and in the pink color, another epitope that is quite amenable to antibody binding and neutralization. So we will hear much more in the coming years about the so-called second-generation HIV neutralizing monoclonal antibodies. But to the extreme right is an antibody that actually has been around for a number of years, which is called ibolizumab. And this antibody, instead of targeting HIV, targets the receptor for HIV, and that receptor is CD4. So the situation is better depicted on this slide. On the top is the virus, on the bottom is the cell. And the viral spike is shown in yellow as the GP120 glycoprotein. And the transmembrane protein is shown in blue as GP41. And many of you would know that HIV enters its target cell by first having GP120 bind to the primary receptor CD4, and then it undergoes a conformational change, and then the virus could engage a second receptor known as CCR5 or CXCR4. And after those interactions, the virus then fuses with the cell using GP41 as the fusion mediator. Now, for the antibody ibolizumab, it actually binds to the second element in human CD4 and somehow prevents HIV penetration into its target cell. And the specific interaction between CD4 and GP120 has been defined for some time at the molecular level to a few angstrom resolution. And more recently, we also understood how ibolizumab binds to human CD4 on the opposite phase of this molecule. And you could see the ibolizumab itself doesn't interfere with GP120 binding to CD4. It actually interferes with the conformational change that is required for infection to occur. Now, I'm going to talk about primarily the use of ibolizumab as an HIV prevention tool. But let me give you a summary first on what has been done with this antibody in the biotech industry. It was originally generated by Biogen as a mouse monoclonal antibody that recognizes the second domain of human CD4 with a high binding affinity and was found to block HIV infection. It later was licensed to Tanox, a former biotech company, and it was humanized using human IgG4 to minimize any effector function. And then it was advanced into HIV treatment setting as salvage therapy or as treatment for people who have very few other options left. And in that setting, it was shown to be powerful in lowering the viral load of HIV through a series of phase 1A, 1B, 2A, and 2B studies performed by Tanox and its successor, a company called TaiMed Biologics. And today it's been shown in 247 individuals or patients without any serious adverse events, some for up to 4 years. And it is poised to move into phase 3 for the purpose of HIV therapy. So here I'd like to emphasize 2 additional points, and that is, first, it is a molecule that's already shown to be active against HIV in vivo and safe in human beings in vivo. And second, for purpose of disclosure, the audience should know that I have links as a scientific founder to TaiMed Biologics. TaiMed is trying to develop this as a therapeutic. However, my research team is trying to advance it into the setting of HIV prevention, something that is quite different. And this prevention program could be summarized in this way. We are trying to take the current antibody as it is and move it toward clinical proof of concept. That is, could it or could it not protect at-risk individuals from HIV infection? And that is a question we want to answer in the clinic. Second, the molecule has its limitations in its current form. And could we make it better? That is, more potent and with better pharmacokinetics. And the answer is yes, and I'll share with you some of those new results. And then I think all of you will recognize that biologics of this sort are expensive, and yet the heart of the epidemic is in Africa, a poorer region in the world, and hence economic considerations are paramount. And so we have to think of strategies that would be affordable for sub-Saharan Africa, and my group in particular is trying to develop a gene transfer method that would allow the body of an inoculated person to make this antibody at in sufficient quantities that would protect such a person and at an affordable price. And this entire effort has been ongoing for a number of years and is entirely, to date, entirely supported by the Gates Foundation in the US. So first, let me talk to you about our clinical development effort. We are already well underway from this timeline chart. You could see that we have essentially finished the phase 1 study. It's a dose escalation study of the antibody given into 3 different groups at higher and higher dosages. The administration is done, and we're doing the final follow-up. And I will simply tell you, without sharing the data with you, that in this study, the antibody has been safe and is well tolerated. There are no local reactogenicity, and we have a pharmacokinetic profile that would permit monthly dosing. And that slide, because of, I think, Mac and PC incompatibility, doesn't show up. But you have to take my word that the PK probably allows monthly dosing with a dose around 500 milligrams subcutaneously. For the past year, we have been doing extensive planning to then move into a phase 2A clinical study in conjunction with the so-called HIV vaccine trials network sponsored by the US NIH. And this would allow us to test more subjects with a chosen dose of the antibody for a longer period of time to obtain sufficient safety data to advance into the ultimate proof of concept study in a 2B trial. And of course, 2A in this setting is a necessary stepping stone to 2B. In the 2B, shown in red, we hope to do as a multinational study in very high-risk individuals, administering either iberalizumab or placebo in a randomized fashion, in particular in high-risk homosexual men. And we envision this as a multinational study that would hopefully include US and China, among others. And for the past year, we've been working together with the Gates Foundation, in speaking with the Ministry of Health in China, speaking with the Ministry of Science and Technology, and about to initiate the same dialogue with the State Food and Drug Administration regarding the possibility of conducting such a multinational phase 2B study. Many of you will recognize immediately that this sets some sort of a precedence because it goes from studies external to China into a phase 2B. But obviously, we want to see, we have a scientific logic and want to see if that could be achieved. Now, you might be asking, why China? Why not do it in US or Thailand in its entirety? And that is feasible. But one of the critical determinant is actually the size of the trial. The size of the trial is obviously dictated by many parameters, including the desired efficacy, including the dropout or retention rates, but another is the rate of HIV infection. And if one looks at homosexual population in the US, the annual incidence rate is probably about 3%. That is, 3% if followed for a year would become HIV positive. But in terms of what's going on in China, the China CDC has been conducting surveillance in many urban centers throughout this country. And I would say nationally, the average would be about 6% per year, 6% per 100 person years. And that is pretty high, and hence would allow us to conduct the trial with a smaller sample size and with perhaps a shorter time frame. In addition, there are a number of investigators here in China and participate in cutting-edge research on the world stage rather than doing Me Too experiments. And so such a study would offer such an opportunity. And lastly, when it comes to the manufacturing of biologics, obviously China, the Chinese organizations are in its early stages. But in fact, there are a number of them now that have the human talent as well as the technical setup to manufacture these products at very, very competitive rates and thereby lowering. And so we're actually in very serious discussion with one such entity in China to manufacture this product for the phase 2B trial and subsequent product if this effort continues to move forward. So let me shift gear and tell you what we're doing in the meantime while these plannings are ongoing. We want to make the product even better in terms of activity against HIV and in terms of its PK profile. First, in terms of activity, iberdomide is far from perfect. And in terms of its antiviral profile against HIV, here shown for 118 strains of HIV, with the black bars reading off the left axis and the red bars reading off the right axis, you could see for maximum % inhibition, there are many viruses that fell short. And in terms of reading the IC50, there are many viruses, particularly on the right side, that have high numbers and therefore are partially or completely resistant to this antibody, even though the antibody is directed to a human protein. And to make a long story short, my research team figured out why those viruses are more resistant. And on this graph, CD4 is shown in green. The envelope protein of HIV is shown in the golden color. And the antibody is depicted in purple and cyan. So heavy chain in cyan and the light chain of the antibody in purple or magenta. And you could see that there is a space between the antibody and GP120, and that space is precisely filled by a sugar, a glycosylation on the GP120, and that sugar is shown in the little balls. And it's as if the antibody works by pushing on that sugar and thereby pushing on GP120. And HIV becomes resistant to this antibody by losing that sugar, by altering the amino acid sequence so it cannot be glycosylated. So we reason that if HIV becomes resistant by that mechanism, why don't we put the sugar on the antibody? And by modeling, we could put the sugar on the light chain in certain critical position of the light chain of the iberdomide. And we could model that with a 7-ring sugar or 11-ring sugar. To make a long story short, we put a large sugar in that position, and all of a sudden, if you look at inhibition, maximal inhibition for the new antibody on the right versus the first generation antibody on the left, if you look at the top panel in terms of maximum inhibition, the numbers are now near the top or at the top. And so maximal inhibition has been achieved. And whether you look at 80% or 50% inhibition, those numbers, the concentration required is much, much lower. So there's no doubt that this antibody is now much better than the original form. I just want to give you another example of how we could make the antibody even better. So we could take iberdomide as it currently is and add on a second antigen binding domain in the antibody field that's known as single chain FV. And that single chain, or SCFV, that single chain FV will allow the fusion antibody to not only bind to human CD4, but at the same time bind to the GP120 of HIV. So it is a dual-pronged attack on the entry process of this virus. And we, in my laboratory, we created a whole library of such bispecific antibodies. And I simply want to tell you that we have one which we call PG9 iberdomide, which is represented in this graph in the right lower panel. And the original antibody is in the left upper panel. And you could see that in terms of maximum % inhibition, this PG9 IMAB is near the top, And in terms of the height of the red bars representing the IC50, they're much, much lower. And this is a log plot, so in general, they're about 100 times lower than the original version. And so that tells us we, at least in vitro, have a better product. And another way of looking at the result is simply to plot the breadth on the top and the IC50 on the y-axis versus what I showed earlier in terms of the more powerful antibodies available in the field. And this new antibody called PG9-IMAP is all the way on the right. And you could see its breadth is 100% and its IC50 is at least one order of magnitude lower and therefore more potent. Another way of looking at that is simply to look at, as you increase the concentration of the antibody, what percent viral coverage would you have against HIV? And you could see this for PG9-IMAP in red. We are operating in a concentration range that is much lower and reaches 100%, whereas the first generation antibody or other available antibodies to date cannot reach 100% and could only reach high inhibition with higher concentrations. So in effect, we are looking at IC50 or IC90 or IC99 numbers in the order of nanograms per ml, again, reflecting the enhanced potency. A number of postdocs in my group have gone on to understand why this molecule confers the enhanced potency. And I will simply summarize it by showing you this slide without sharing with you all the technical experiments. In effect, what this fusion molecule is doing is, and the fusion molecule here is shown in both blue and red, and it is binding, it is anchoring on the receptor shown in green. And when it anchors on the receptor, it shuts down the receptor. At the same time, it concentrates the red element at the very site where HIV is trying to enter the cell. And so the increased concentration of the red moieties, which would then bind to the red circle on the HIV trimer, is enormous. And hence, its potency is elevated by approximately 100-fold. The other thing we're trying to do is simply to improve the PK. Because this is an antibody that is directed to a human surface protein, it is subject to endocytosis and degradation as that receptor molecule is recycled. But the human cells will remake human CD4, but of course, ibolizumab will be consumed. And there are a number of tricks in the field that could be applied. We have already carried out two of these three things. First, we changed the IgG4 to an IgG1 to make the molecule more stable. Second, we have modified the Fc region, that is the bottom portion of the antibody molecule, so that it is recycled much more efficiently via the so-called recycling receptor known as FcRn. And then we are now changing the antigen binding region with histidine substitutions that will allow the antibody to release from the receptor in a more acidic environment of the endosome. And when that happens, the molecule would be better recycled. So these things that we're trying to do, hopefully, will give us a 10-fold improvement in the pharmacokinetic profile of this molecule. With the two modifications already made, we know that we could achieve about a 5-fold improvement in monkeys, in animals. let me conclude by just showing you this slide. We are trying to also express the antibody in vivo. And this could be done by, because of many technological improvement in DNA inoculation. So we could improve the vector, we could improve the enhancer and promoter elements, and we could deliver it more efficiently into the muscle via electroporation. And when one does this, you could see in the experiment shown on the right, we could actually give it every few months and achieve concentrations, in this case in a mouse, that is way above what we need. Now the question is, in a larger animal setting, in a monkey and ultimately in a human, could we do the same? And that is a effort that we hope to be able to finish up in about 3 years' time. And such a methodology of delivering antibody would be orders of magnitude cheaper and more affordable than administering antibodies that are made by current biotechnology methods. All of this, ultimately, we hope to be able to achieve proof of concept for iberalizumab in HIV prevention in humans using the current first-generation molecule. We hope to make a second-generation molecule with the profile that I have listed on this slide, and then at the same time see if we could come up with a more affordable delivery strategy. So let me finish up by saying that all of this would not be possible without a very talented group of colleagues and collaborators. I'd like to recognize a few in particular. Huang Yaoxing has been working with me for many, many years, and he's actually originally from Shanghai. And Craig Pace, another colleague, and Song Ruijiang, another. And our clinical effort is advanced by a combination of Marty Markowitz, Katrine Myers, and Neil Pate. In China, I've been working very closely with my colleague at Tsinghua named Zhang Ningqi, and also Professor Sang Hong in Shenyang and Professor Wu Hao at Youan Hospital in Beijing. We've been supported by Ministry of Health, particularly Liu Chen, Liu Buzang, and also by a number of key officials within the Ministry of Science and Technology. This effort is also supported by the Gates Foundation. Two important officials in Seattle, Nina Russell and Francine McCutcheon, and in the Gates China office by William Xiong. And all of this would not be possible without the collective contribution of all of these individuals. Once again, I thank you for the opportunity to participate in today's conference, and it's a great honor for me to be one of the keynote speakers.
下面请让我介绍今天的第二位 keynote speaker, Dr. Alberto Guignono. 和这个我们的何医生不同 ，Guignono 博士可以说是一位在药物的后期开发以及注册上市方面有着非常丰富经验的专家。目前 Guignono 是在 Parexel， 也就是金鼎咨询公司任全球的副总裁，并且负责 Parexel 在日本的日常工作。Guignono 博士是在美国的杜克大学取得了学士学位，然后在North Carolina 北凯罗来纳州大学取得实验心理学的博士学位。他曾经任美国药物法规事务学会董事会主席，并且是在任 DIA 董事会2000年到2004年的董事会成员， 也是 DIA 2007年全球大会的主席。他今天演讲的题目是21世纪新药全球市场的开拓，协作与融合。下面请我们欢迎他。
Good afternoon, everybody. Great to be back in Shanghai at this very important conference, and thank you all for being here and making it so successful. What I would like to do today, with your permission, is to focus on the practical implementation of two fundamental concepts, and those are collaboration and convergence. Collaboration, of course, is on the very title of this conference, and convergence, I think, is what today's collaboration is all about, as we all attempt, with great energy, to bring new medicines to global markets in the 21st century. I would like to begin with some very broad and high-level considerations, and they have to do with ideas that were generated at a very recent workshop on scenario planning held by the DIA Board of Directors in March. I have DIA's permission to share these with you, and fundamentally I begin with these because they're very big picture considerations that will drive all of our lives, particularly in the healthcare arena. There are great uncertainties in the next 10 to 15 years about what will happen in the healthcare world. We don't know what is going to happen. But within the DIA, we thought about this for a day and a half, and we've come up with a few ideas. And what I'm going to present is just a few slides, 4 or 5, about divergent outcomes in the healthcare world. And none of us are really sure what is going to happen. But these are some thoughts for your consideration and reflection, not only today, but in the coming months and years. The first one has to do with health and wellness, and who will own health and wellness? Who will be accountable for the health and wellness of the world's people? On the left-hand side of the slide, will the system take care of us? Will the system, whatever it is, provide total coverage, reimbursement, regulation of health care? Or on the right hand of the slide, will the patient ultimately be responsible and accountable for his or her own health and well-being? Will the patient be in a position to self-determine his or her health and wellness, take personal initiative, change lifestyles, instead of expecting that the system will take care of the patient in every case? And if we have such a scenario where the patient is ultimately responsible, will there be a growing gap between the rich and the poor, those who can afford health care and those who cannot? Another phenomenon that could have divergent outcomes, geopolitical power and influence. Will the world be a stage where there is shared influence, where developed markets and developing markets will share the stage, where, in fact, trade agreements will govern relationships among economies in a very macro way? Will there be global enforcement of product safety and quality? Or conversely, will there be domination by one or a few countries driving innovation and trade? Will there be neo-isolationism across the world, with each country basically retreating within its own boundaries in every way? Global drug development, this is close to our interests, of course. On the one hand, will there be a lot of regulatory convergence? More about the concept of convergence a bit later in my talk. Will there still be global clinical studies driven by multinational firms? Will there be a focus on global diseases, on global health and patient information? Will there be a lot of real-world studies? Or will there be basically a lot of regulatory fragmentation, with each country and economy basically focusing on its own regulatory system? Will development be driven by small firms? Will national studies, local clinical data be uniformly required? Will there be very few global collaborations? These are very divergent potential outcomes. Health information technology, a huge development in this century. On the one hand, will there be a lot of protection of private data? Will genetic information of individual patients remain private? Will real-world data availability be very limited? Will local data basically stay within each country and not leave that country? Or are we going to have a very open world where, in fact, there will be relatively little or no privacy of healthcare information? Will there be global data banks? Will researchers, doctors, patients have access to huge data from the real world? Will government use data of such nature to drive policy decisions? We don't know. But these are dilemmas that we live with today. And finally, therapeutic paradigms. On the one hand, are we going to see healthcare systems delivering health to large populations in a uniform way, treating populations rather than individuals, where the individual becomes less important as long as large populations benefit from treatments? Or will there be more individualized therapy, where the patient really is the focus of each therapy, where in fact genetic subtypes, personalized medicine, the right drug for the right patient at the right time, and companion drug and diagnostics will be the way that we develop new medicines? Will the patient be increasingly empowered to make own decisions and to discuss individual needs with his or her doctor? And will we be able as societies to stand and tolerate the high cost of medicines that are basically targeted at very small subpopulations, very few numbers of individuals? These are, again, true dilemmas. And I don't know the answer, certainly, but these are some critical uncertainties that we're facing today. There's certainly the obvious. There's rising global demand for unmet medical needs. These are the therapeutic areas where, in fact, the medical needs are quite significant still in the 21st century. And at the same time, we see that the pharmaceutical industry revenues are growing at a slower pace than in the first 5 years of the 21st century. We also see R&D investment by pharma companies flattening, growing at a lesser rate. So if we basically see this reality and realize that the industry is facing slower growth in sales and the slower growth in R&D spending, then industry and its partners must focus on what I would call intelligent innovation that will basically minimize the risk of failure of new medicines in development, and will try to maximize global market access because there is less money to invest. Therefore, each investment must be more intelligently targeted to produce the desired outcomes in terms of new medicines for the world. There are barriers to global market access. It's nice to talk about global market access, but there are significant barriers to it. And these are just three, and I'm sure you can think of many more. One is the failure of drug development. Secondly, local regulatory requirements. Thirdly, what I would call sponsor, regulator, and payer misalignment. Let me comment on these briefly. First of all, failed drug development. Let's realize that about half of phase 3 clinical studies actually fail to show what they're intended to show. The failure rate for phase 2 studies is even higher. We might expect that because in phase 2 we're still seeking proof of concept. But phase 3s are our largest, most expensive, most global, in many cases, studies, and they fail at about a 50% rate. The reason for that ranges from safety concerns to efficacy, failure to show efficacy, to CMC issues and others. And about half the time, the problem is related to the dose. We don't do a good enough job of determining the appropriate dose of a drug. About half of the FDA complete response letters, which are fundamentally letters sent to sponsors saying we're not ready to approve your new drug application for the following reasons, half the time the issues are related to dosing. The sponsor failed to explore the full dose range, or the lack of safety exposure data in the proposed dose range was the case, or there was a failure to show that higher doses provide any greater benefit than lower doses. With respect to local regulatory requirements, I'm going to show you a couple of slides from Jerry Stewart of Pfizer on behalf of the Pharma Asia Pacific regulatory team as he presented them in 2011. And fundamentally, the challenge for the regulatory strategist is, number one, to secure marketing authorization of product X in as many markets as possible, as rapidly as possible. The mechanism these days is essentially to run global phase 3 studies of drug X in the US, Europe, Asia, and Latin America, and to do so with a relatively reasonable patient enrollment. The rules often require that for local countries, the data in local patients must be provided. So local clinical data are still required in a number of countries. And that causes a challenge and forces a compromise in study design between the sponsor and any number of countries. So the way to achieve global drug development comes in at least a couple of forms. One form may be phase 1, phase 2, and phase 3 studies that are basically country-specific or region-specific. The other way is to do global phase 3 in all the regions of the world at the same time to try and accelerate. However, we do then run into those local regulatory requirements. So this pharma committee basically suggests, guess what? Collaboration. as well as compromise. A communication pathway to regulators that basically ensures that there is an understanding of what phase 3 programs must look like and must be like. And if compromise is going to happen, compromise should be achieved in such a way that the development of a new important medicine can be achieved with a minimum of inefficiency and a maximum of speed. To the third point I was trying to make, sponsor-regulator-payer misalignment. What I mean by this is that sponsors, the regulators who are in charge of approving or not approving a drug, and payers who are in charge of reimbursing or not reimbursing a new medicine are sometimes not aligned. And here's an example. We've all heard of NICE, the UK National Institute of Clinical Excellence. And here's a quote from the Wall Street Journal, May of 2011. In another sign of growing opposition to high drug prices, NICE said Britain should not pay for a group of expensive leukemia drugs for certain patients, saying their cost outweighs their benefit. This is, of course, after the sponsor has developed the medicine and the regulators in the UK have approved it. The payers refuse to pay. Another example, this drug was rejected by the UK National Institute for Health and Clinical Excellence, which suggested the company consider lowering the price of the skin cancer treatment because it could not be considered to be cost effective. There's no way to identify which patients will benefit from the drug. So these are examples I would suggest of misalignment, and a greater cooperation among sponsors, regulators, and payers would perhaps solve this misalignment. I'll come back to that later. Let's pause a minute for the word collaboration, which is in the title of our conference and in the title of my presentation. Collaboration does not mean we must all agree on the same thing. It actually means leveraging similarities where they exist. It means accepting legitimate differences where they exist. But it also means converging on a common goal. And that's why today I like to talk about collaboration and convergence. Let's talk about an example of collaboration that doesn't result in identical outcomes, and that is decisions made by the FDA in the US and the European Medicines Agency. In an analysis performed by the FDA on new molecular entities that have been approved or not approved since 2006, the analysis shows that 63 new molecular entities were approved by both FDA and EMA. 21 were rejected by both, but 11 were approved by FDA, and 14 were approved only by EMEA and not FDA. What does this say? It says, number one, these two agencies are in communication, and we know this because they say that they communicate all the time, very frequently. But it also says they have their differences of opinion. They don't reach the same conclusions on the same drugs with the same data. They come at this from different points of view. And this is a fact of life. It must be accepted. It is not just a phenomenon of these two agencies. It happens all over the world. But these two agencies and others are committed to the public health. They come at it from perhaps different points of view, but the commitment to improve the public health is common to these two and every other regulatory agency. So collaboration does exist. Identity of outcomes does not. I'm an optimist by nature, and I'm going to offer an optimistic view of the future, uncertain though it is, and we have to live with those critical uncertainties. But I would like to share with you some thoughts based essentially on 4 trends that we have observed as impacting the regulatory environment in the next 10 or 15 years. And they are the trends of innovation, convergence, efficiency, and quality and safety. I'll take each of these in turn. Number one, innovation. I do believe in innovation, and we are blessed today that Dr. Ho basically showed us what innovative minds can achieve when thinking differently about outsmarting an extremely clever virus. This is a wonderful story, and we can all feel very inspired, I certainly do, about what science can do for us and for the world's health. So drug discovery, translational medicine, personalized medicine are phenomena that are still evolving in real ways. There are examples of success. I think there will be many more examples of success in the future. As a result, clinical development will evolve to facilitate the transformation from discovery to medicines for patients in very novel ways, with novel study designs, enrichment designs, adaptive designs, and many others that perhaps we have not yet devised. Drug and diagnostics will be co-developed and co-approved in greater increasing numbers. There will be faster roads to approval resulting from true innovation, because regulators will respond to true innovation that changes the practice of medicine. Transparency of clinical data will reduce and eliminate barriers to global competition. An example of innovation in personalized medicine, a drug that FDA approved in 3 months for a mutation in cystic fibrosis. Only 5% of patients with cystic fibrosis have this mutation. In those patients, this drug works beautifully. In the other 95%, it does not. And yet, FDA was very excited to approve this drug. It is an example of personalized medicine. We wish we had many more such examples in the next 10, 20 years, in half century, to show how patients with specific diseases can be treated effectively and safely with new, clever, and cleverly designed therapies. Science, I believe, must lead the way. Molecular biology is leading us to a deeper understanding of diseases. Personalized medicine has been represented by a number of recent approvals by various regulators with examples of medicines that are targeting specific genetic subtypes. Ethnic factors are still with us. There was a workshop yesterday on ethnic factors in drug development. There's still a concern in many realities, in many economies. But we're transitioning gradually from geography to biology, where in fact biology and a deeper understanding of the pathophysiology of disease and the biology of individuals will go beyond geography, and I dream of the day where the expression foreign data will become a thing of the past. We will not talk about foreign data anymore. We will talk about the biology of the individual patient who is about to be given one or other medicine depending on his or her condition, genotype, phenotype, and other circumstances. We've also in this table is from ICH E5 guideline, the extrinsic factors and the intrinsic factors. Maybe someday this table will also become obsolete if science leads the way. A Japanese investigator, Professor Tokin, has conducted studies comparing PK/PD profiles among Japanese, Chinese, Korean, and Caucasian normal volunteers and found no differences with the 3 drugs listed here. That gives us reason to hope. On the other hand, the Japanese PMDA points to data for other drugs where in fact there are important differences due to so-called ethnic factors. These are the realities we have to live with today because science hasn't yet taken us to the next level, to the next step. PMDA also likes to show this slide indicating that for 1/3 of the drugs approved for marketing in Japan, the US dose was more than 2 times higher than the approved Japanese dose. And this is a concern to the Japanese authorities, whose job it is to promote and protect the health of the Japanese people. So one can certainly understand this point of view, and once again, we call on science to help us decode and understand the way that we should go. That was innovation. And again, innovation, I think, will lead the way, will make it possible for the rest of us to develop medicines for the world's health. But moving to the second trend, convergence. I believe that regional regulatory collaboration will increase, especially in the emerging markets where such collaboration has not yet become mature. Asia and Latin America will see greater convergence of local regulatory systems. We already see some of that happening, but more, I think, will occur. And regulatory systems will come to resemble one another more and more. They will never be identical. There will be always local priorities, but the resemblance will be such that 20 years ago we could not even have imagined it, and even today we strain to think what it might look like, but I believe it will happen. Let's talk a bit about regulatory convergence in more depth. You may remember the Common Technical Document. It did not exist 20 years ago. The regulators in Japan, US, and Europe got together. Under ICH, they produced over 60 guidelines in the last 21 years. One of them was the Common Technical Document Guideline. And what a wonderful example of convergence. This is the photograph of the steering committee and the Common Technical Document Expert Working Group within ICH. All the folks that contributed to those guidelines and the CTD creation. And we know the CTD. It almost points to convergence as a triangle with the vertex at the top. And it was a way in which regulators and the regulated could decide, yes, we can converge our documentation, we can put it in a common format except for Module 1, we can in effect make things easier for both the industry and the regulators. And the Common Technical Document has in fact achieved success. Now, other countries beyond ICH use the CTD. Other countries use a form of CTD. The ASEAN CTD has 4 modules, not 5, but it works fine. It's another example of convergence put into work and made to work. And again, an example of thinking creatively and making sure that what needs to happen does happen. There are other harmonization initiatives that perhaps were inspired by ICH, perhaps by other needs, and they're listed here, and they span the globe, from Asia to Latin America to Africa. The 14th International Conference of Drug Regulatory Authorities said many things. One of the things it said was collaboration and cooperation among regulatory agencies is important. Most recently, the East African Community held a meeting at the end of March, attended by some in the audience, including DIA management and the President. And basically, they called for greater cooperation in East Africa, in the countries of Tanzania, Kenya, Rwanda, Burundi, and Uganda. And basically, they also feel the need for greater cooperation and perhaps convergence in an era where the world's regulators and the world's industry are fighting very hard on the basis of strong science to bring better medicines to the world's people. APEC, the Asia-Pacific Economic Community, basically has the Regulatory Harmonization Steering Committee as one of its initiatives. And it now uses the word convergence with increasing frequency. This is a slide from FDA's Justina Molzan, and regulatory convergence is defined in this group as representing a process whereby regulatory requirements across economies become more similar or aligned over time as a result of the gradual adoption of internationally recognized technical guidance documents and standards. It is not harmonization, it is convergence. Convergence towards a common goal, which is global health. As a result of this concept of regulatory convergence, this group within APEC is basically focusing on projects that will address and are addressing multi-regional clinical trials, good review practices, product quality, pharmacovigilance, and many other initiatives. Again, an example of regulators and regulated working together to achieve something that is meaningful for the world. There's also a phenomenon, it's a quadripartite consortium involving Health Canada, Swissmedic, the regulators in Switzerland, HSA in Singapore, and TGA in Australia. These slides were provided to me just last week by Swissmedic with permission to share them with you all. And what this consortium of 4 countries is attempting to do is to pool resources to achieve a common goal. The initiative started 6 years ago. And the basis was a network of bilateral agreements between pairs of these regulators. They've since then held regular meetings, quarterly telephone conferences of the heads of the 4 agencies. They have a detailed work plan. They're starting by sharing information. And the idea is to have early access to important safety information for the protection of their respective people. They've gone now from information to work sharing. The information sharing is ongoing. The work sharing is a little bit newer. And they are doing parallel reviews. They've completed some pilots. They obtain prior agreement from sponsor applicants to share these parallel reviews. They exchange assessment reports. They analyze and compare their findings. And they reach independent decisions, much like FDA and EMA have done and continue to do. These 4 agencies will reach independent decisions. But throughout the process of reviewing a drug's application, they actually communicate and share work practices. They share the work. They are attempting to be more efficient in the way they do things, in the way they do their work. So the specific work-sharing initiatives include new chemical entities and biologics, generic drugs, good review practices, assessment of benefit and risk, and participation in ICH. So in conclusion on this consortium, it is a good basis to establish long-term cooperation, initially by sharing information and then by sharing drug reviews. The principal fields of cooperation have been well defined. The final goal is to share work, thereby reducing the workload for a single agency. This ought to be very attractive for regulatory agencies that are small and understaffed. And there are many of those in the world. It may be that most regulatory agencies in the world are in fact small and understaffed. So this example of convergence among regulatory agencies, of collaboration, could perhaps be a model for other agencies to follow, to pool resources, to do their work more efficiently, while continuing to promote and protect the public health of their constituents. There's a model of tripartite cooperation right here in East Asia that I'm sure you're all familiar with. China, Korea, and Japan have been in these tripartite talks since 2007. They've had periodic meetings. The last one was in Tokyo last November. The next one will be here in China sometime in 2012. And their thoughts and initiatives have included research on ethnic factors, information exchange on clinical trial regulation, and most recently, the drafting of a China, Japan, Korea regional clinical trial guideline, which we hope to see soon. So the idea in this tripartite dialogue is to see how these three agencies can collaborate productively in this very important part of the world. The next two slides simply show PMDA's view of how it is possible to leverage multi-regional clinical trials for drug approvals. These are two PMDA slides that basically show that PMDA has approved 19 drugs for Japan on the basis of multi-regional clinical trials. So this can happen and does happen. There is a lot of cooperation and dialogue in many parts of the world, including, for example, across the Atlantic and the Pacific. The red line basically shows EMA-FDA communications on a monthly basis in the last couple of years. The blue line at the bottom shows PMDA and MHLW communications with EMA. They could probably become more frequent, and I think the intent by PMDA is to make them more frequent. But the point is, these communications are occurring today. They're not a hope for the future. They're occurring today. And they, I think, provide hope that collaboration and convergence will become more common. My own reflections on this topic are that a reduced insistence on local clinical data, if it happens, is going to potentially accelerate drug development and market access of important new medicines. Can this be achieved? Can less insistence on local clinical data actually be achieved? A second reflection is that in Europe, which is 27 member states of 500 million people, does not require local national clinical data before approving a medicine for marketing in Europe. So the question in my mind is, can East Asia, with 2 billion people and fewer countries than 27, consider a similar model and perhaps converge its requirements on the basis of considerations that we could all reflect about and are not for today? Two is to combine and integrate regulatory strategy, clinical strategy, and commercial and pricing strategy at the same time, and understand what it is that we need to do to develop a medicine that regulators will approve and payers will pay for patients all over the world. And the ultimate goal, the roof of this building, is to achieve the global launch of practice-changing, meaning drugs that change medical practice for the better, drugs that actually prolong the cancer patients' lives, not by months, but by many years or decades, drugs that revolutionize healthcare and make it possible to defeat the diseases of today. That is what practice-changing innovation is. That innovation must in the end be reimbursable to patients, it must be available to patients who need them, must be appropriately priced, must be affordable. So how do we do all of this? There must be firstly an internal convergence within pharmaceutical companies, I would suggest to you. In order to achieve this reimbursable innovation, regulatory affairs, the clinical people, and the commercial people must think at the same time in the same room. Traditionally, this has not happened. Traditionally, regulatory and clinical have collaborated to a large extent. Commercial has been off to the side. And these two groups of people have not really been very comfortable communicating for a long time. They're now beginning to communicate and integrate their activities and thinking and planning in novel ways that even 10 years ago did not happen. And the goal of this effort is to generate data that are focused on regulators and payers. And those data must be planned for and collected during drug development, not after drug development, as was often the practice. There's also a different type of convergence that needs to occur outside pharma industry, and that is convergence between the sponsors and the regulators on one side and the payers on the other side. And these three constituents must agree, design, and deliver demonstrated efficacy, safety, and clinical value to patients, because it doesn't make any difference if regulators approve and then the payers or the system that is supposed to provide drugs to patients refuses to pay for them. The goal is reimbursable innovation. Innovation that is not reimbursable ultimately does not benefit the patient. Medicines must be practice-changing. They should have either a broad impact on the public health or produce wonderful outcomes for individual patients who have no other choice. Regulatory approval is no longer sufficient for commercial success. It used to be sufficient back in the 20th century. But today, a reimbursement strategy that is based on sound, innovative, strong science, on compelling medical innovation, on excellent drug development, must be accompanied by a reimbursement strategy that sees the payers at the end of the process interested in reimbursing these important medicines, interested in paying a fair price, interested in making these medicines available to patients. But payers cannot be left as an afterthought. The payers must be integrated into the conversation early in drug development, not at the end. They must be sitting down with regulators and sponsors to have these discussions. There already are examples in Europe where EMA is holding meetings with both sponsors and payers, health technology agencies, HTAs. This has happened now for many months. It is a productive conversations. They may not always be successful, but the dialogue among regulators, sponsors, and payers has in fact started, and the road has now been traveled. The third trend, and I will close in a few minutes, the third trend is efficiency. I think the best regulatory practices will in fact become more common across regulatory systems across the world, and of course across companies as well. Regulatory science is a relatively new concept that is beginning to drive more and more regulatory agency decisions. Science is fundamentally at the center of these decisions, and agencies that are able to apply regulatory science to their decisions are doing their constituents and their patients a big favor. There will be also, I think, improved regulatory infrastructures with competent regulators, efficient regulators, and structures that are appropriately sized for the markets that they seek to promote and protect. And therefore, I think that the efficiency gap between developed and developing regulatory agencies will gradually become very narrow and non-existent as technology will help us all become more efficient, as communication becomes more streamlined, as decisions are based more and more on regulatory science and strong data. The fourth trend, quality and safety of products, a paramount concern to all of us as patients and as consumers. Enforcement and regulatory pressures will increase. We've all seen problems with product quality and safety in recent years. This will not go away. Enforcement is going to rise. Manufacturing in particular will see greater and greater scrutiny. We see that today. This will increase. And regulatory demands for truly real-life pharmacovigilance, addressing product safety concerns, will increase as well. The world's largest regulatory agency, the United States FDA, has admitted it cannot do this alone. In their report for last July, titled Pathway to Global Product Safety and Quality, they basically included these 4 points. FDA will do the following things. It will create and participate in global coalitions of regulators. It will leverage global data information systems and networks. It will engage in intelligence gathering and intelligence use. And it will perform risk-based leveraging of combined efforts of government, industry, and public and private sector third parties. They don't mean the United States. They mean across the globe. FDA has basically said, we're such an interconnected economy that even we, the largest agency in the world, cannot do this alone. They are promoting and asking for collaboration with other regulators. This is a huge signal in favor of collaboration and convergence. And to demonstrate that they're serious, they've engaged in collaboration with EMA and PMDA and a number of companies in a consortium on international serious adverse events. A very important initiative, of course, and again an example of collaboration. So the future regulatory landscape, in my opinion, will in effect broadly follow innovation. If the innovation is sufficiently irresistible, if in fact the science brings us medicine ideas and potential drugs that are going to be so compellingly effective and safe that regulators really can't not approve them, we will see greater speed and efficiency in regulation and approval of new medicines. And regulators in most regions of the world will in fact adopt practices that will facilitate market access, not slow it down or even block it. But science and drug development have got to deliver very important innovation for the world's patients. Finally then, my message is that a collaborative convergence is now necessary and is upon us among sponsors, regulators, and payers. Specifically, sponsors retain the responsibility to innovate, to invest in science, whether it's academic science or their own intramural science. Sponsors will continue to generate drug development data, will continue to manufacture and look after product quality and distribution and safety monitoring. The regulators, imbued with scientific competence, regulatory science, able to provide sound scientific advice, in possession of adequate infrastructure to do their job properly with sufficient resources. Certainly inspecting, certainly enforcing will be an important collaborator in this enterprise. Last but not least, the payers, the health technology agencies, have an obligation to be transparent in how they decide whether to reimburse or not reimburse a new medicine. They need to be fair and realistic. They have to keep patient benefit and value in mind, as they do. They need to be efficient. They need to communicate their thoughts about cost effectiveness, and they must share with sponsors and regulators early in drug development what is their vision for this medicine. Will they be willing to reimburse it and make it available to patients? What are the roadblocks from the payers and reimbursement community? Will they stand in the way of new medicines, or will they facilitate access of these new medicines? And if these three parties collaborate and converge, as I'm suggesting, I believe we will deal with a healthcare world that is even better than what it is today on the basis of very strong illuminating science. Thank you very much.